Hello, a warm welcome to all of you in our 3 days online certification course on topic molecular docking. In last lecture, we discussed how we prepare the protein for the docking purpose. Okay, so in today's session, we understood how we perform the docking and how we interpret the result of docking. Okay, so we start the session. So first, in last lecture, we already download this protein and we already download this ligand. Okay, so first I open Pyrex. Okay, this is the interface of Pyrex. Now here first we load the protein. So load the molecule, and in last lecture we already discussed how we clean the proteins. So that particular clean protein is present in my document. This is the protein. Now this is the protein. This protein is PDF format. So I convert this, right click, then autodock, then make as a macromolecule. Now click on this one. Now our protein is ready. Okay. Now I load the ligand. For loading the ligand, click on this open label, then this one, plus sign, then in downloads, this is my ligand. Now I open this ligand, right click, then minimize the energy. Okay. Now convert this into the autodock PDT format. Okay. Now our edit the previous one. Now this is the ligand. Okay. This is the ligand file. This is my previous ligand files. Okay. This is the target. This is the previous one. Works. Okay. This is not a new one. So I close all this. Okay. This is your this is the ligand. This is your protein. Now click on this Vina wizard. Now click on start. Here one ligand is already selected. One macromolecule means protein is also selected. Now click on forward. Okay. Here this is the and this is the to set the grid. Okay. Let's suppose if you perform any specific docking or you want to perform the docking at a specific position, then you also maximize or minimize this grid board like this. Okay. Accordingly you set. This is for the central, this is for the dimension. Okay. You already, by removing this, you also set your grid. Okay. But here I perform the blind docking. So I simply click on this maximize. Okay. Means I cover in my protein if at OR position ligand. Now here I perform the blind docking. That's why I minimize. I set this grid box in maximize position. Okay. Now click on forward and wait for the result. Within 2 hours, within 2 or 5 minutes, you will get your result. Okay. Running. Now this is the result. Okay. Now our docking is completed. This is the result in analysis result. Okay. Our affinity result is literally very good. Okay, this is affinity is minus 9.4, minus 9.2, minus 9.1, 8.2, 8.8.9, 8.8, 8.7. Okay, these are. Now we understood this is the binding affinity in calorie per mole. This is the mole, means this is the position. Where this is the zero. Yes, I will show you this. Okay, this is the RMSD value. Lower the RMSD value, more the appropriate result. This is the RMSD RMSD lower bound, and this is for the upper. Okay, less the value, more the appropriate result. Means RMSD means root mean standing. Okay, RMSD value, RMSD means root mean square deviations. Okay, this RMSD value, what is the RMSD value? I will explain you detail. Okay, now for downloading this, simply click on this one, then download in your desired folder. Let's suppose my desired folder is this one. Now DNA barcode, molecular docking, and I name scoring result. Let's see and save. Okay, now this result is saved in my PC. Okay, now we understood how many models and what is the exact position. So simply click on the protein, then molecule, click and display. Click here, then click on this plus sign, then open this one, then here and display. Now this one total 9 models. Okay, now I removed out this protein. These all are the ligands, means actual poses of your ligand. This is the now I remove. I will show you one by one. Okay, this is the 0. This is the model number 1 of your ligand. Okay, now open. Okay, now I show you one by one. Now I disclose all these poses first. Okay, now this is your macromolecule. This is the size. Okay, this is the first position of your ligand. Here is a clearly shown. Okay, now this is the second position. This one. Okay, model number 2. This is the now this is the model number 3. Okay, pose basically. This is the fourth. This is the fifth. This is the sixth, seventh, eighth, or ninth. Total 9 position. Means this is your active site. All the protein ligands are bind at this position. Okay. Now I save this model number 0. Model number 1 or this one. Model number 0. Okay. And this is the result. This one. Okay. So for downloading this, simply click Save as PDB format. In my desired folder is this one, molecular docking, and this is the model number 1 and save. Now ligand, your protein is downloaded in your PC, ligand is downloaded in your PC. Okay, now open this Discovery Studio. Okay, then file, then open. First upload your file in PDB format. This is my protein. Okay, this is my protein. Okay, now open. This is my ligand. Okay, now copy this ligand and paste here. Okay, now we check the interaction. So simply click on ligand interaction. Now this is the interaction, means this is the position via which ligand is bind our protein. Okay, so what do you want to interpret? Aromatic atom, click here. Now these are the aromatic atom. Okay, you want to know the hydrogen bonds, click here. Now these are the hydrogen bonds. This purple is for the donor and this green is the acceptor. Okay, you want to find out the charges, click here. These are the charges. You want to find out the hydrophobic nature, click here. This is the hydrophobic nature, basically hydrophobicity. You want to find out the anagen availability, then click here. SRA, click here. Expand, contracts. Okay, here if you want to find out the distance, click here. This is the distance. Show type, what are these? Distance as well as types, different types of bonds. Okay, if you want to expand this interaction, click here. Will interaction will expand. Contract. Okay, okay. Pocket of atom, this is your pocket atoms. This is your ligand, this is your interactions. Ligand. Now you want to see the receptor, click here. This is the receptor. Okay, here these are the, if your protein in your protein number of ligands is present, then you simply find out the information about the ligand by using these options. Okay, this is for the experiment. Okay, now here if you want to see the 2D interactions of your protein, then click here. Okay, this is the 2D confirmation of your ligand key. What is the ligand, how many amino acids are participate, each and every informations are available here. Okay, here what is the van der Waals interactions, conventional hydrogen bond, pi sigma bond, amide P strict P amide, complete information are available here. Okay, superposition proteins, here the number of options are available. This is for the view interactions, this is for the define the binding site. Okay, and this is for the superposition and superimposed protein. Okay, I click on this view interactions, means this is your ligand, this is the 3D interactions view, means by the help of this you easily understand how in 3D form how the protein is bind your ligand is binding your protein, and this is your model and this is the 2D information. For downloading this, click here file, save as 3D interaction. In which format you want, these are the number of options. I want the graphical representation and save. Okay, and same for this one, file, save as. This is the 2D interactions and save. Okay, 3D, 2D, and here the receptor, ligand, pocket size, number of information are available. You will get the information according to your requirement. Okay, so the diagram, this. Understood? In this way, you easily find out the each and every parameter by the help of Pyrex as well as Discovery Studio. Okay, so this is the way by which we easily perform the molecular docking. Okay, first this is the way. In Pyrex, we simply load the ligand as well as protein and convert this in PDBQT format and find out the interactions. Okay, and now open this folder, molecular docking, and this is my scoring result. Okay, this is the scoring result. Ligands, their position, their RMSD value. Lower the RMSD value, more the appropriate result. Means more the appropriate, this ligand is more appropriate. Okay, here this value is a little bit similar, minus 9.4, 9.2. For interpret the steps of data, we go through the hydrogen bonding. Okay, but here the RMSD value is 0, 0. It's more appropriate, means more less the RMSD value, more the appropriate result. Okay, in this way you easily perform the molecular docking by the help of Pyrex. Okay, if your system is not supporting the PDBQT form, sometimes Pyrex. Okay, in this way you easily perform the molecular docking by the help of Pyrex. Okay, now we understood how we interpret this data. We also now we understand what is this RMSD value. Okay, RMSD value full form is root mean square deviation. Okay, and by the help of this RMSD value we easily find out the average distance between the uh, we easily find out the okay, we easily find out the average distance difference by the help of this RMSD value. Okay, okay, by the help of this RMSD value we easily find out the average distance average distance difference average difference between a 3D molecules. Okay, less the RMSD value, more the appropriate result. Okay, Okay, if your RMSD value is in between to 0 to 1 angstrom, if this RMSD value is if this RMSD value is 0 to 1 angstrom, it means it is excellent match. Okay, excellent match or almost perfect overlap. If this value is 0 to 1 angstrom, it's mean good or reliable. If this is value between the 0 to 3, it's moderate. But this value is more than 3, it's not acceptable, it is poor result. Okay, in this way we easily interpret the result by seeing this RMSD value. If the RMSD value is 0 or 1, it's more appropriate. Okay, in this way you easily predict the result of molecular docking and easily perform the molecular docking. Okay, this is all about the today's session. In the today's session we discuss about how we perform the molecular docking in Pyrex. Okay, and how we interpret the result. Okay, here the RMSD value and binding affinity plays a most important role. Less the affinity, more the appropriate result. Less the RMSD value, more the appropriate result. Okay, and this is all about the today's session. Thank you so much. Now we discuss how we predict the protein structure by the help of AlphaFold. Okay, okay, so. So now we understand how we predict the protein structure. For predicting the protein structure, there are two ways. First is your experimental method and second is your computational method. Okay. In computational method, again there are three methods. First, your structure is present in database. Database like UniProt, like PDB, like Sysprot, etc. Next, homology modeling. Homology modeling means if a same structure is present, then you similar template is present. What is present? Similar or known template is present, then you move to the homology modeling. Homology modeling. If your protein structure is completely unique, then you move to the ab initio method. Okay. In ab initio method, you use AlphaFold, iTracer, T-Rostra, etc. Okay, now we understand how we predict the protein structure by the help of AlphaFold. Okay, this AlphaFold is a very heavy software. Okay, for running the software we require a very high speed processor. Okay, and normally at the education level it is not possible. That's why we choose a alternative method. Here in a tool that is Chimera, in Chimera option is present, that is the structure prediction by the help of AlphaFold. So the algorithm behind this tool is same, all the procedure is completely same, but tool is different. Means here we use Chimera for predicting the protein structure by the help of AlphaFold. Okay, algorithm is same, procedure is same, all the procedure is same, but tool is different. Okay, so now we start how we predict the protein structure by the help of this AlphaFold. Okay, so first in Google, first in Google you simply search AlphaFold database. Okay, then open this first link and here you search your target protein. Okay, like here I search your target protein like dehydrogenase. Okay, then this is the result here, reviewed or unreviewed. Okay, and this is the average PLDT score, and this is the population. This is means dehydrogenase enzyme related to the Homo sapiens, mus musculus, whatever. Okay. Okay, these are the popular organisms. If you want to search the dehydrogenase related to the humans, so simply click on Homo sapiens. Okay, same for the other organisms. Here, for searching the molecule, you simply apply the filter. Now, these all are the result related to the Homo sapiens. Okay, now let's suppose I want to this one. So I click on this one. Now this is the protein. This is the glycerol 3 phosphate dehydrogenase met. This is the glycerol 3 phosphate dehydrogenase from mitochondria. Here this is the protein summary. This is the gene, that is the series GDP2 gene. Organism is a Homo sapiens uniprot, and this is the primary structure of this protein, and this is the structure. Okay, here this blue regions means the confidence level of this structure is more appropriate or very high. Okay, here this is the graph. If your confidence level is very high, then it will show in a blue color. Okay, if the confidence level is high, means rank is between to the 90 to 70 to 90, then it will show in the sky blue color, this portion. If your confidence level is low, it will means your LDT value is between to 50 to 70, then it will go to the under the low level. If your LDT value is very less, means less than 50, then it means your confidence level is very low. Okay, in this reason, this blue color is means high confidence level. High confidence levels means your LDTT value is more than 90%. Sky blue reason, this one. This reason means why? Because the LDT value is high, means between to 790 to 70. Okay, it is acceptable. But this this yellow color means your LDT value is between to 70 to 50. It's mean very low. Okay, but this red color is very low, means LDT value is very less, means below to the 50, and it is not acceptable. Okay, in a graph, this is the aligned residues and the expected position error in angstrom. Okay, more the green region, more the appropriate result. Okay, this graph is showing between the this graph is plot between the scored residue and aligned residues. Okay, in this graph, more the yellow, more the value, more the green color, more the appropriate result. Okay, and this graph tells the expected position error. Okay, if in your error, if in your protein in your protein, if light green color is more, then it's mean your protein is not appropriate. If in your graph more the red, more the green color, it's mean your structure is more appropriate. Okay, now here your average LDT value is 86.25%. It's mean it is acceptable. Okay, now this is the information about the domain. This is the sources, cat, quality, boundaries, length, domains. Okay, these are the different domains, domain 1 and model confidence level. Okay, domain 1, domain 2, domain 3, domain 4. Okay, all the domain information are available on this page. Next, in annotation information is also available. Okay, now this is a very most important point in this structure. These are the similar proteins, okay, which have a 29% similarity, 28.5% similarity, E values, their expected residues, their E. coli, all the information are available here. Let's suppose we choose this one, this first one. means this protein have a 29% similarity with our sequences. Now I will take this protein. Okay, I will take this PDBID. Okay, now in next step, I open Chimera. Now this is the Chimera X. Okay, in this protein, in this model, this is the interface. This is the home page of the software. Here the number of options are available. This is for the file. Here we also upload our protein. This is the option for editing. This is the option for selecting which view you want. You want to select the functional group, protein, nucleic acid elements. Okay. This is for the chain. This is for the residue. This is for the structure, sequence. Number of options are available here. This is the action. Action, atom, cartoon structure, references, color, labeling, etc. These are the tools. Here we also find out the binding analysis, and this is the structure analysis. For structure analysis, this is the structure editing, structure prediction. For structure prediction, we use a AlphaFold. Okay, and these are the favorites, presides, helps. Okay, number of options are available. Here, I simply use a command for opening our target protein, and so simply write open and name of our target protein template, sorry. Okay, that is the 2RGO. Okay, so simply write 2RGO or enter. Okay, now this is the protein. Okay, in this protein there are two chains is present, A chain or B chain. Here a non-standard residue is also present, that is the FAT. Okay, now I maximize this. This is the structure, and we also change this structure by using different options. Here this option descent, snapshot. Okay, spinning. This is the for spinning option. Okay, if you want to spin the protein. Okay, now I stop this. Okay, this is the protein. This is for the hiding or showing. Predicting the structure of our target protein, you simply go to the tools, then structure prediction by AlphaFold. Okay, whenever you click on this one, then these are the options shown. First is the fetch. Fetch is for open the database structure with most similar sequences. Okay, search means by the help of this we easily find out the similar sequences in AlphaFold database. Okay, if the similar structure is present in AlphaFold, then the structure is showing in this box. Predicting means if your structure is completely unknown, then you move to the prediction part. First we click on fetch. Okay, now sequence search found one AlphaFold ID do not have AlphaFold database model. Okay, now means no similar proteins is present related to this A chain. Here I select only one chain. Here this option is also present to change the chains. Okay, now search. No model is present. Okay, means this structure is completely unknown, means our giving sequence is completely unknown. So simply click on predict. Whenever you click on this predict, then loading, then this interface is opening. Here, not cancel. Here you simply paste the sequences, your desired sequences, and simply click on this one, run away. Okay, whenever you click on this one, then this start software is automatically start the running procedure, and it will take approximately 1 hour. Okay, and here for one email address you can run this software 2 hours at a day. If the time is increases, then you move to the subscription part. Now this is the result. Okay, this is the algorithm behind this. I minimize this. Okay, this is the algorithm behind this. Okay, this is the algorithm behind this. In this software, first install your AlphaFold Collab software, then it will run this command, and after running this command it will create this sequence coverage and identity graph. Then it will create a total 5 structure. First 5 structure in different angle. This is the first angle, second angle, third angle, fourth angle, and fifth angle. And this software also predict your aligned error graph. Okay, this is the result. In this way, you easily predict the tertiary structure of protein by the help of this AlphaFold. Okay, let's suppose uh, you have a new sequences and you want to predict the structure. So here you simply, this is the position. You simply copy this, delete this, then paste the sequence in the sequence bar and predict your structures. Okay, in this way you easily predict the tertiary structure by the help of this AlphaFold software. Okay, and in this structure, this structure is completely aligned. This blue color is showing key means this is the structure that is predicted by the AlphaFold. Okay, and here this is the best model. Now I select previous one. This is the structure that is predicted by the AlphaFold. Okay, more the blue color, it's mean the structure that is predicted by the AlphaFold is more appropriate. Okay, showing, this showing. Okay, if you want to select this one, hide. Okay, in this way you easily predict the tertiary structure of protein by the help of this AlphaFold by using this Chimera X software. Okay, okay, in this way you easily predict the protein structure by the help of Chimera X. Okay, in this software, first you use AlphaFold. In this way you easily predict the you easily predict the tertiary structure of protein by the help of this AlphaFold. For this, first you open AlphaFold database, then search your target protein, then find to the similar PDB ID of your target sequences, then load this in Chimera X software, then click on this tool, then structure, then click on this AlphaFold, and the software automatically predict the structure. Okay, it will give the total 5 structure on different angles. Okay, in this way you easily predict the tertiary structure of protein by the help of AlphaFold. Okay, so now we already discussed how we perform the molecular docking. We already discussed how we perform the molecular docking by the help of Pyrex. Okay, so now we discuss how we perform the molecular docking by the help of online software. Online software like CBDoc2. Okay, so we start today's session and okay, so we start how we perform the molecular docking by the help of this CBDoc2 software. Okay, so in Google you simply search CBDoc2, open this first link. Okay, this software is a cavity detection guiding the blind docking. This software is for this web tool is for the blind docking. Okay, so now click on this get start. Okay, stay away. Now here this is the two options. First, here you upload your protein, here you upload the ligand. In my system number of proteins are available, so I simply click on this one, choose. Now in downloads number of proteins are available. Let's suppose I choose this one. Okay, this is the protein. Now I'll choose the ligand. So let's suppose I choose this one and choose this one and open. Okay, now your ligand is uploaded. Now I already upload the protein. Let's suppose I choose this protein. Okay, now my ligand is already uploaded. Sometime it will showing the issues, so simply rename your ligand and upload here. Okay, this is the ligand, this is the protein. In this protein 64 heteroatoms, all not the water molecules, is automatically deleted by this web tool. And okay, and these are the ligand is present. If you want to perform the docking by the help of this ligand, so simply choose and start. But I want to dock with this one. Okay, Now I click on this search cavity. 98% completed. Now click on this view result. Okay. Now this is the result. Okay. This software detects total 5 cavity. Okay, cavity number 1, cavity number 2, cavity number 3, cavity number 4, cavity number 5. Okay, cavity means here is the position where the ligand will bind. Okay, more the volume, more the appropriate cavity. Okay, okay, this is the center and this is the cavity size. Okay, now this software automatically choose the 3 cavity. If you want to choose manually, then you simply choose or de-choose this one. Okay, this is for the center, this is for the full screen, surface, hide the pocket, show the pocket, hide the label, show the label. All the options are available. You choose these options according to your requirement and click on this blind document. Now this is the result of document. Okay, this cavity 1 shows the minus 8.9 Vina score. It means this is the more accurate result. -5, -4 is not appropriate. -8.8 is more appropriate. Means if the Vina score is less, means affinity is very good. Means first interaction shows the best cavity. Okay, this is the cavity side, this is the axon, this is the docking side, and this is the residue which are participate to bind with this ligand. Okay, if you want to download this one, then simply click on you also download the ligand is mol2 format, PDB format, and as well as you also download this protein and ligand in PDB format. Okay, now this is for the center. Now I have to maximize this. This is for the center. This, by the help of this, we easily change the first I change the receptor style in backbone. Now this is the ligand, this is your receptor. Now I change the ligand. Okay, and this is the ligand uniform. Receptor y space filling, y lower layers, y surface, y cartoon. Okay, and we also color by chain, by secondary structure, by hydrophobicity, by elements, by beta factor, by uniformity. Okay, this is the result. For downloading this, simply click on PDB. Whenever you click on PDB, then this will download it in your PC. Okay, this is the result of docking by the help of CBDoc. Okay, so in this 3 days online workshop, we easily understood how to perform docking by the help of Pyrex, by the help of CBDoc2, and we also understood how we predict the tertiary structure of protein by the help of AlphaFold. Okay, and this is all about today's session. Thank you so much for watching my video. Thank you so much.